Good afternoon, good morning, and good evening to everybody. Greatly appreciate everyone taking the time to attend our 2025 R&D Day presentation. PeptiDream, our R&D efforts are focused on 5 core therapeutic areas: radiopharmaceuticals, oral peptide therapeutics, peptide-Oligo conjugates, Peptide-Cytotoxin conjugates, and multifunctional peptide conjugates. And across these 5 core areas, we are focused on creating a robust clinical pipeline of innovative programs. In today's presentation, we will touch on the current status of these efforts and the exceptional progress we've made across these areas in fiscal year 2025. On slide 4 is an overview of our current radiopharmaceutical clinical pipeline. In 2025, we had an exceptional year with our Curium PSMA partnered program entering registrational trials in Japan, and the first wave of PeptiDream discovered peptide RI conjugates for both collaboration efforts and in-house efforts progressing forward. With our Glypican-3 program partnered with BMS and our FAP programs partnered with Novartis, we had 4 programs entering phase 1 studies. We also recently filed INDs for our wholly owned in-house CA9 programs. Additionally, our second in-house Claudin 18.2 program, announced last year, is progressing through IND-enabling studies, while we in parallel are planning a phase 0 imaging study. And today, we are very happy to announce our third in-house program, targeting cadherin-3. These results are the product of the last 5 years of discovery efforts, and these programs represent just the first wave of programs to enter the clinic for us. We have a robust preclinical pipeline of peptide RI programs following, and we're on course to move more programs into the clinic in 2026 and beyond. So it really represents a very, very exciting time for us in the radiopharmaceutical field. Today, we'll start off providing more detail on our efforts in the radiopharmaceutical space with our CMO and also president of PDRadiopharma, Masato Murakami, highlighting the exciting progress and efforts happening at PDRadiopharma. Murakami-san.
Thank you, Patrick. So, hello, everyone. And I'm Masato Murakami. So, I'll briefly introduce our company, PDRadiopharma. We have been Japan radiopharmaceutical leader since 1968 and advancing nuclear medicine for society and delivering the new era of the radiopharmaceutical impact to the patients. First, our positions. We are Japan's only domestically owned pure-play radiopharmaceutical company with more than 50 years of continuous experience in the nuclear medicine. Over that time, we have built nationwide manufacturing, logistics, and technical support that are difficult to replicate. And this gives us a unique role in Japanese ecosystem. Second, our commercial foundations. We already have a profitable cash-generating portfolio of approved nuclear medicine products. This stable base allows us to invest in new growth areas. Finally, our future growth drivers. Our mid-term strategy is centered on radiopharmaceutical, with some launched product and late-stage assets that I will walk you through today. In particular, Amyvid is now entering a multi-year, high-visibility growth phase and will be one of the key contributors to our next stage. Next slide, please. Today, I will walk you through 5 key highlights for fiscal year 2025. First, Amyvid, our amyloid PET tracer. Second, Riot MIBG for PPGL and neuroblastoma. Third, the kappa ATSM program in malignant glioma. Fourth, our kappa and lutetium PSMA INT program in prostate cancer. And finally, our total nuclear medicine solutions. I will touch on each of these. Next, please. Let me start with highlight number 1, amyloid. The key message here is amyloid is putting amyloid PET at the center of Alzheimer's care. And we are driving adoption of amyloid PET as a standard tool of the treatment decision and follow-up. In other words, amyloid is evolving from the diagnosed tracer into a critical piece of modern Alzheimer's treatment pathways. Next, please. Over the past few years, Alzheimer's disease has changed dramatically. Antibody therapy, such as lecanemab and donanemab, have made disease-modified treatments a clinical reality. But they require accurate patient selection and response assessment. In diagnosis, A-beta tracer imaging with amyloid is now in routine practice, allowing physicians to visualize amyloid beta plaque directly and to evaluate how patients respond over time. Together, these advances are moving the field from simply seeing the disease to active treatment and monitoring A-beta. Next, please. On this slide, I'd like to highlight 3 growth drivers for amyloid. First, the launch of the lecanemab and donanemab are expanding the pool of the patients who require amyloid confirmation and on-treatment monitoring. Second, our AMICRUISE and AMIFLOW2. These applications provide standardized and quantitative PET assessments, including centiloid scale scoring, which supports consistent treatment decision and follow-up in daily clinical practice. Third, PET imaging offers direct visualization and quantification of the brain amyloid distributions. These 3 elements together support steady structural growth in amyloid utilizations. Next, please. This slide shows how the demand is translating into real numbers. In 2024, amyloid sales were roughly 7 times higher than 2023, mainly driven by reimbursements and the first wave of the disease-modifying therapy use. In 2025, we expect about 2.5-fold additional growth versus 2024, as both lecanemab and donanemab roll out, and PET becomes standard for the treatment initiation and follow-up. And importantly, 2026 is positioned for continued high double-digit growth as 12-month monitoring scan after therapy initiation. This became routine. So amyloid entering the multi-year high visibility growth phase, generating increasing cash flow for PDR. Next, please. So highlight number 2 is the Riot MIBG. Here's the story about the transforming care in PPGL and pediatric neuroblastoma. Riot combines imaging and therapy and has evolved from one of the earlier nuclear medicine pair into a modern standard for rare endocrine childhood cancers. I will touch on both of the medical value and our effort to expand access in Japan. Next, please. Riot MIBG consists of 123 MIBG for imaging and I-131 MIBG for treatment. This matched pair enables precise patient selection, treatment planning, and then target irradiation of the tumors. Riot is indicated for MIG-avid unresectable pheochromocytoma and paraganglioma, and more recently for MIBG-avid neuroblastoma. It built on decades of the clinical experience in Europe and the US, but only recently became available in Japan, closing the long treatment gap for this rare but devastating disease. Next, please. Slide 14 brings this to life with a pediatric case example. On the left, you see the child with high MIBG uptake in metastatic neuroblastoma, especially multiple bone marrow metastatized before the treatment. On the right, marked reduction in tumor uptake after the Riot therapy. For children with high-risk neuroblastoma in Japan, Riot changed what was previously a very limited treatment landscape into a real, accessible therapeutic options. For family and pediatric oncologists, it means moving from hoping for MIBG to actually being able to deliver MIBG therapy in Japan. Next, please. Of course, approval alone is not enough. Access is critical. MIBG therapy has been used overseas for many years, but Japan approved Riot for PPGL in 2021, and then for neuroblastoma in 2025 this year, after roughly 30-year drug lag. This is a rare cancer, not a high-volume market, but it is a high-impact commitment to patients with endocrine tumor and childhood cancers. We are therefore working to expand the number of the certified MIG treatment centers across in Japan. The solid circle on the map shows current size, and the open circle shows planned expansion next year. Our goal is simple. No child who needs Riot MIBG should miss the chance to be treated in time. Next, please. Highlight number 3 is kappa ATSM in malignant glioma, a program led by our partner LinkMed. This radiopharmaceutical targets hypoxic tumor microenvironments and has already advanced into the phase 3 registrational trial. For PDR, this program represents a disciplined way to participate in a first-in-class opportunity while respecting the clinical development driven by our partners. Next, please. Here, you can see the program overview and phase 1 STAR-64 results. Kappa ATSM is a small molecule radiopharmaceutical that selectively accumulates in the hypoxic tumor tissue, a key feature of the recurrent malignant glioma, where prognosis remains poor and treatment options are very limited. In the phase 1 STAR-64 trial in Japan, total 18 patients were treated across multiple dose levels. The maximum tolerated dose was identified at 99 millibecquerel per kg, with no serious treatment-related adverse events, showing the drug was generally safe and well tolerated. In glioblastoma patients, median overall survival reached 17.7 months, with 1-year survival of 64.8%, compared to historical expectation of around 30 to 40%. This is very encouraging data. Therefore, authority PMD allow to start the step 64 phase 3 registration trial directly. Next one, please. Step 64 is a randomized phase 3 study comparing kappa ATSM to best physician choice in recurrent high-grade glioma. Enrollment is ongoing, with overall survival as a primary endpoint, and filing in Japan anticipated around late 2027. In this program, Link met lead clinical development and overall strategy. PDR is in charge of the regulatory strategy and filing in Japan, and we are preparing domestic manufacturing and distribution infrastructure to ensure timely supply once product is approved. This structure gives PDR exposure to a potential new therapy in malignant glioma, while also showing the win-win partner-oriented approach, where each party focuses on each strength. Next, please. Highlight number 4 is kappa and lutetium PSMA INT. And this is for prostate cancer, developed together with our global partner, Cunningham. Here's a story about building a scalable radiopharmaceutical platform that combines PET imaging and target radiotherapy to reach the large PSMA positive patient population. I will first touch on the market opportunity and then on our development strategy. Next, please. Prostate cancer is the second most common male cancer worldwide, and the prevalence is expected to roughly double between 2022 and 2040. More than 25% prostate cancer express PSMA, often at the level 100 of the times higher than normal tissue. On the left, you see global PSMA PET imaging market projected to grow from about $1.5 billion in 2024 to $3.5 billion by 2033. On right, the PSMA target radiotherapy market is expected to grow even faster, from $1.4 billion to over $6 billion in the same period. Clinically, radioligand with high sensitivity and specificity are enabling earlier detection and better disease monitoring. And PSMA PET is now incorporated into the NCCN guideline as a core imaging modality. At the same time, lutetium-based PSMA therapy have shown strong anti-tumor activity in mCRPC, establishing PSMA radiotherapy as a new treatment options. Next, please. This slide illustrates where PSMA target radiotherapy fits along the prostate cancer journey. With PSMA target radiotherapy, eligible patients, the post-ARDT pre-taxane mCRPC segment is a large and fast-growing group. Patients in this setting typically receive multiple cycles of the PSMA radiotherapy, which translate into the substantial and durable radioligand demands. This means we can effectively serve this segment. It provides long duration, high value opportunity for our lutetium PSMA INT program. Next, please. Finally, let me explain our development strategy in Japan with Cunningham. Globally, Cunningham is running the phase 3 trial that provides a core efficacy and safety dataset for both imaging and therapy. In Japan, we are pursuing a compact, capital-efficient approach. For kappa PSMA INT, we are conducting an open-label, multi-center study in newly diagnosed prostate cancer, aiming to position in both as a standalone diagnostic and as a companion imaging tool also. For lutetium PSMA INT, a single bridging study in post-ARDT mCRPC is designed to leverage together. And Cunningham positive phase 3 ECLIPSE results, which have already met their primary endpoint, kappa imaging and lutetium therapy, supported by small Japanese bridging trial, and PDR nationwide infrastructure, create a differential PSMA platform, not just a single drug, and measure medium to long-term growth engine for PDR. Next one, please. So highlight number 5. And this is our radiopharmaceutical ecosystem. Beyond individual product, we are building the defensible end-to-end platform that combine radiopharmaceutical, hardware, software, and service. This strategy deepens our relationship with hospital and make it easier for new sites to adapt nuclear medicine. Next one, please. Delivering nuclear medicine safety and at the scale require far more than innovative compounds. On this slide, you can see our total solution platform. A portfolio of diagnostic and diagnostics, radiopharmaceutical, injection system, dispenser system that automate complex workflow and improve safety, software solutions for image assessment and radiation dose management, and information system that support compliance with international standards. By combining these elements, PDR lower barrier for new sites, improve efficiency and safety for the staff, and enhance the patient experience, while at the same time building long-term strategic partnership with hospital. This ecosystem, together with pipeline, I have just described, position PDRadiopharma to lead the next era of the radiopharmaceutical innovation and growth. This is from my side, so I hand over to Patrick.
All right. Thank you, Murakami-san. Next, I will discuss partnered and in-house discovery and development efforts in the radiopharmaceutical space going on at PepTiDream. On slide 26, as I mentioned previously, in the last 12 months, these are both discovered programs at PepTiDream in collaborations with Novartis for FAP and Glypican-3 for BMS, what was Raised Bio and has become BMS. On the left, the FAP program, which is related to a variety of different tumors. This is overexpressed on fibroblasts in a variety of different types of solid tumors. The program FXX489 with the therapeutic using lutetium and the diagnostic using gallium entered into a phase 1 clinical trial earlier in the year, the end of 2024, early 2025. Novartis highlighted some of this work at an oral AACR presentation in 2025, highlighting the program as a best-in-class potential for radioligand therapy, a program we're very excited about. On the right side is the Glypican-3 program, which also initiated a phase 1A, phase 1B study earlier in the year. And this product is being developed for hepatocellular carcinoma, liver cancer, in a wide range of patients. Again, being run by BMS, and we're very much looking forward to future updates to come for these 4 exciting programs, 2 diagnostics and 2 therapeutics. PD32766 is our first in-house peptide RI program. targeting CA9, carbonic anhydrase, which is highly expressed in the majority of clear cell renal carcinoma patients. We have previously highlighted the strong preclinical evidence for this program in selectively targeting CA9 and showing clear anti-tumor efficacy in animal models. To further de-risk the program, we coordinated with the National Cancer Center Japan to run a physician-led imaging study using this copper-64-based diagnostic in patients with ccRCC, referred to as a phase 0 study. The study, conducted in just a few patients, demonstrated clear CA9 tumor targeting with strong tumor to background ratios. We saw no dose-limiting or unexpected safety signals, and these findings provided us validation in humans of the tumor targeting ability of our CA9 program, both potentially as a new diagnostic for kidney cancer, but also potentially as a powerful therapeutic. These results really gave us confidence to take this program forward into the clinic ourselves. We conducted IND-enabling studies since last year through much of 2025, culminating in the recent filing of 2 INDs, IND for the diagnostic and an IND for the therapeutic in the US. We are on course to initiate the phase 1 trial in the US in 2026. The phase 1 trial, similar to most targeted RI compounds, will consist of a phase 1A dose escalation part, where we image patients for CA9 positive tumors to enroll to receive the therapeutic agent, which we then give in escalating doses to confirm tumor targeting, dosimetry, and safety. And from this data, we will select the recommended best dose to take forward and expand patients at that dose in the 1B portion, or the dose expansion portion of the study, to better clarify therapeutic activity as well as pharmacokinetics and safety. We are very much looking to get this study underway in 2026. On slide 30, we announced our second in-house program at our R&D Day last December 2024, targeting Claudin 18.2. Claudin 18.2 is a member of the Claudin family of tight junction proteins. In gastric and pancreatic cancer, the growing tumor causes cell morphology changes that result in the exposure of Claudin 18.2, making it accessible for a drug to bind to and target. We leverage this biology to create PD29875, a peptide RI conjugate targeting 18.2. Similar to the CA9 program, we're developing a diagnostic therapeutic pair, and so we'll be able to identify patients with the diagnostic agent that would most likely benefit from receiving the therapeutic agent. At our 2024 R&D Day, we shared the exciting preclinical data showing strong tumor-killing efficacy in animal models for this program. At current, IND-enabling activities are ongoing and progressing on schedule. We are additionally, of course, planning for a phase 1 study in patients with gastric and/or pancreatic cancer in the future once those studies are completed. In parallel, we are again coordinating with the National Cancer Center Japan to run a phase 0 imaging study to generate some initial human tumor targeting validation to both de-risk the program and also assist with planning the most efficient phase 1 study we can do. So very excited about next steps for our 18.2 program as we head into 2026. This year, we are happy to announce our third in-house peptide RI program targeting cadherin-3. Cadherin-3 is a member of the cadherin family of cell adhesion proteins. Its overexpression of cadherin-3 is strongly associated with epithelial mesenchymal transition, or EMT, where epithelial cells lose their structured characteristics and acquire mesenchymal traits. EMT is a major driver of cancer metastasis, allowing cells to break away from the primary tumor, colonize, distant organs. Cadherin-3 is highly expressed in head and neck squamous cell carcinoma, as well as cervical cancer, lung cancer, and a variety of other solid tumors, making it an attractive target for a targeted peptide RI approach. Our cadherin-3 targeting peptide RI conjugate shows high affinity and selectivity for cadherin-3. It shows specific accumulation in tumors expressing cadherin-3. And when chelated to either therapeutic lutetium-177 or actinium-225, shows strong tumor killing efficacy. All of which, as a preclinical package, supports our decision to announce this program as our third in-house program and take this program forward into IND-enabling studies going forward. That concludes the radiopharmaceutical portion of our presentation today, and we will next move on to our 4 non-radiopharmaceutical focus areas, of which we will provide various highlights on each of these. On slide 35 is an overview of our current non-radiopharmaceutical clinical pipeline. In subsequent slides, we will touch on some of the exciting advances we have made across these areas in 2025. I would just note on this slide that for the 2 Merck programs in phase 1, we remain limited in what we are actually allowed to say about these 2 exciting programs. We'll start off initially with our peptide therapeutics and oral peptide therapeutics efforts. A major clinical highlight of 2025 was seeing Alexion advance our GHR program, LXN2420, into a global phase 2 study. The study is being run in 11 different countries across a large number of sites, testing LXN2420 in combination with somatostatin analogs in adult patients with acromegaly. The study, of course, follows the previous phase 1 study completed in 2024 by Amylite Pharma before they were acquired by Alexion AstraZeneca, that showed excellent safety and dose dependency in reducing and maintaining IGF levels in healthy volunteers, which bodes very well for the success in this ongoing phase 2 study. We're very much looking for the future results around this program as it progresses forward. We also had our sister company Peptide complete a phase 1 safety study of PA001 in healthy adult and elderly volunteers. PA001 was found to be both safe and well tolerated and exhibited excellent pharmacokinetics. And we are now considering next steps for this program to potentially address an unmet medical need around long COVID and or elderly individuals that still have certain challenges. As we mentioned recently in our announcement earlier in the week, partnering activities remain ongoing for our oral myostatin program. While these activities have been ongoing, we have continued to build a strong preclinical data package supporting the program. In 2025, we ran numerous additional animal studies showing that our peptide alone could actually induce lean body mass growth by itself, and that when given in combination with semaglutide, animals lost more fat and less muscle compared to semaglutide alone. We also showed clearly that our peptide has a unique dual mechanism of action, and that the peptide binds myostatin, and the peptide myostatin complex can still bind to ActR2, but when ALK is recruited for signaling, the peptide blocks that surface on myostatin, and no downstream signaling can be sent through the SMAD pathway. But because the peptide-bound myostatin is also capable of occupying the ActR2 receptor, it is possible that it is therefore also blocking the activin signal itself indirectly. This unique dual mechanism of action is significantly different from the other biologics out there targeting this pathway. Also in 2025, we conducted additional safety studies, oral absorption work, human dose prediction, synthetic route analysis, and COGS assessments and optimization, further adding additional data to the already strong package. This program is progressing toward IND-enabling studies as they continues to drive this program toward the clinic, while we continue in parallel our partnering efforts and activities. Next, I'm going to hand it over to Christian to present more of our non-radiopharmaceutical program efforts.
Thank you, Patrick. This is my second year as Chief Scientific Officer at Peptidream, and as you've seen, we've had an incredibly exciting year and significant progress across our late-stage and clinical portfolio, including the addition of our third RI-PDC development program, and obviously, as Patrick just discussed, the continued progression of our myostatin inhibitor program towards GLP tox in parallel with partnering discussions. Throughout the year, I've had an excellent time working with our development teams across our R&D organization to really drive on an exceptional year for both our internal and collaborative preclinical portfolio. Now I'm going to provide updates on the remaining core focus areas of Peptidream, starting with our oral peptide therapeutic updates, other than myostatin. For us, oral peptide therapeutics represent a very strong area of future growth. As you know, peptides can achieve similar or better efficacy as compared to a biologic, and their much smaller size facilitates better tissue penetration as well as target engagement, delivering much more efficacious approaches. Importantly, by delivering our compounds orally, we are able to enable combination therapy approaches in many indications where single treatment alone is not enough to see complete responses. Peptides offer synthetic manufacturing, and that route of administration allows very easy access, especially in chronic disease settings where patients may be taking these daily for the rest of their lives. This is incredibly important, especially in immunology indications, in terms of this field where patients live with some of these diseases for their lives. The synthetic nature of these also offer greater shelf life stability and reduce costs associated with cold chain storage, enabling wider access for many patients. Here at Peptidream, we've now been leveraging our rich collaborative experience, as well as our strategic investments in AI, ML, and next-generation PDPS technologies that I spoke about at last year's R&D Day, to really unlock high-value oral macrocyclic peptide therapeutics. And with this, I'm truly excited to announce our second wholly owned oral peptide therapeutic program targeting IL-17, a clinically validated and high-value target for the treatment of psoriasis. As you may know, psoriasis affects just over 41 million patients worldwide, which accounts for 2 to 3% of the world's population. The current market is valued at about $28 billion as of last year, with IL-17 biologics earning about 30% of that value. There are numerous indications in the immunology and inflammation space that inhibitors of IL-17 can be truly a radical treatment for our patients. But importantly, the current offerings are biologics that are administered by subcutaneous injection. And therefore, oral delivery is highly desired for these chronic disease treatment and these combination approaches. Our proprietary and wholly owned macrocyclic peptide is not only an inhibitor of IL-17A, as is done with Cosentyx and Taltz, but also inhibits IL-17FF, similar to the biologic Bimzelx. Clinical data has shown that inhibition of all three forms of IL-17 are important for the depth and durability of response in moderate to severe plaque psoriasis. Internally in our preclinical research, we have shown equivalent efficacy to a commercially available biologic in an inflammation mouse model, as compared with our peptide being orally administered in that same model. We also see greater distribution and tissue penetration into the skin, as compared to antibodies, where over 24 hours our peptides are located almost twice as high in skin as they are in plasma, compared to antibodies, which show a significant reduction in that tissue accumulation as compared to plasma. This offers us a significant differentiation over current biologics. And we are very excited about this program and are rapidly progressing this program into IND-enabling studies moving into 2026 to accelerate clinical entry, while we are also exploring the potential partnership opportunities in parallel. Moving on to our next target focus area is peptide-oligo conjugates. Oligonucleotides are a very fascinating class of drug modality, as they have the potential to drug the undruggable targets through not only, instead of going after the protein side, but targeting the expression and degradation of the mRNA that then enables their translation. This therapeutic area also enables the pharmacoevolution against mutations that can be rapidly adapted due to the fact that this is a genetic-based medicine. There's been a lot of success in this modality over the last several years. There are now 6 approved RNA-based drugs using siRNA or ASO technologies. However, all of these therapies are targeted to the liver. The next frontier in this space is being able to deliver oligonucleotide therapeutics to target tissues beyond the liver. The benefits that we see by using PDPS-discovered peptides is to enable this precision targeting through their ability to generate high affinity and specific peptides against tissue-specific targets, enabling also the speed of discovery to enable rapid in vivo proof of concept, as well as then having the fast systemic clearance and exposure to reduce things like immune activation and off-target toxicities. Importantly, as mentioned, our peptides and this modality is enabled through chemical synthesis, allowing easy modification of the linker and payload stoichiometry. A really exciting aspect about this platform is that when we have tissue-specific peptides, each peptide can truly enable numerous therapeutic applications, as we can attach a variety of different cargoes in order to tackle some of a variety of different diseases, as shown here. And we have several disclosed and undisclosed tissue-specific collaborations with a variety of large pharma and biotech. Importantly, at PeptiDream, we have our own suite of peptide-specific molecules that target specific blood-brain barrier carriers that are also available for the licensing on a specific payload basis, allowing us to get at truly unmet medical needs in the neuroscience space. And we do expect some very exciting announcements on our oligo delivery portfolio coming very soon. Moving along, I'll touch briefly on our cytotoxic PDC platform. As you know, there has been a significant amount of investment and growth over the last several years in the antibody-drug conjugate space. Currently, there are 19 ADCs approved worldwide, with over 200 ADCs in clinical development, targeting over 50 different antigens. Last year alone, there were 27 R&D partnerships signed, totaling $9.4 billion. However, antibodies also have significant liabilities, including long systemic circulation, on-target and off-tumor toxicities, as well as complex manufacturing, which are still significant hurdles for this field. We believe that the peptide offers significant differentiators as compared to an antibody modality for delivering these cytotoxic cargoes, including enabling the high affinity and selectivity towards tissue targets of interest, that smaller size enabling tumor penetration, but also importantly, higher payload delivery per dose, and that fast PK reducing that off-target toxicity while maintaining tolerability. Renal clearance offers us that reduction of hepatotoxicity, and again, by being synthetic, we can actually play with things like linker, payload, as well as stoichiometry with ease. We also expect to have very exciting announcements on this portfolio to share with all of you in 2026. And finally, as the last focus area, I will speak to our multifunctional peptide conjugate platform. We've had some very exciting and significant preclinical data generated internally around this, making this a very exciting exciting area for growth in the future. Really, the idea behind this is to replace the next generation multispecific biologic area. There are currently 16 bispecifics with over 600 candidates in clinical trials right now. As you can see on the left, there's a wide variety of formats that are being used in order to try and get at this multispecific functions, leading to what can lead to very complex manufacturing. At PeptiDream, we have created a variety of proprietary novel linkers, allowing us to utilize peptides that we have already discovered across our variety of different platforms in order to really engage this platform in terms of conjugating these together and enabling us to generate multispecific peptide conjugates in rapid speed. The data we have been generating this year is allowing us to form a platform so that we can actually begin to start thinking about how we are going to move these programs internally, but also for potential partnering opportunities in 2026. And with this, I'll turn it back to Patrick.
Thank you, Christian. On slide 51, as we wrap up our 2025 R&D Day, I just want to highlight that 2025 was simply the most exceptionally productive year for us in our history. We had 12 clinical portfolio transitions. You saw a number of programs that we've been working on for some time in the preclinical setting advance into phase ones, programs move from phase one into phase twos, and also partner or in-license programs moving into registrational studies in Japan. It was simply a transformative year as far as in regards to our clinical pipeline. As we focus across all 5 of these modalities, at current, we have clinical candidates for 3 of these modalities, but we expect to see the Peptide-Oligo and Peptide-Cytotoxin areas also yield clinical candidates in the near future. These efforts are the result of years of hard work by all of the people here at PeptiDream, by all of the team at PDRadiopharma, and of course, across all of our fantastic and phenomenal collaboration partners. I fully expect that this progress and the successes of 2025 are going to carry over to 26. And going forward, this robust clinical pipeline that we've been working to recreate will simply expand and grow further. As a closing, I just want to end today's presentation with all of that excitement, of course, is the rationale for us to invest further in PeptiDream and PDRadiopharma. We have high confidence in the business, and we've undertaken 2 large capital programs here. On the left is building PeptiDream 2.0. This is at our currently existing site, where we hope to double the research R&D available space, adding new state-of-the-art functions to our already existing facilities. This is progressing nicely through the design phase. You can see a nice illustration on the lower left of how this looks, and we're very hopeful that we'll have shovels in the ground in 2026 to get this underway. On the right is a new manufacturing facility for PDRadiopharma for us to launch in-house radiotherapeutic programs. This is located in Chiba, strategically located between Haneda and Narita Airport to give us the best domestic coverage and allow these exciting products to be brought to hospitals and patients across Japan. Additionally, this is actually progressing very, very nicely through the design phases, and we also hope to get shovels in the ground in the coming year. So 2 big programs for us. These are both extremely motivational for employees across both organizations to see that the successes we're having in our R&D and efforts are translating, of course, into company growth. So with that, I'm very happy to take any questions and very much look forward to the future for us.